Okay, folks, it looks like it is the start time. It's right at 7 o'clock here this evening. So I just want to thank everybody for joining us tonight for the Wheat Streak Mosaic webinar. And you'll see that we have it labeled Fight the Mite. And so we're going to talk a lot about the wheat mosaic viruses and the mites that vector those viruses. And this actually webinar came about actually this spring and early summer when we were getting lots of calls about wheat streak mosaic and those types of symptoms showing up in fields, and a lot of questions surrounding what made 2025 such a wheat mosaic year. And so as we were talking through this, Lucas Haig, myself, Kelsey Anderson Onifree, as we were talking through this, we said, maybe we just need to have a big effort to get information out in front of folks. And so one of those efforts is this webinar this evening. So thank you everybody for joining us. We'll kind of go through what we're going to talk about this evening. And so we're going to talk about wheat streak mosaic virus, triticum mosaic virus, and high plains mosaic virus. All of this we call the wheat mosaic virus complex because all three of these look very similar in the field, show up in fields about the same time, and are all vectored by the wheat curl mite. So that's really going to be the focus of our talk tonight. So our schedule this evening is we're going to start off with our wheat streak mosaic complex and the wheat curl mite biology. So Kelsey Anderson Onofree, who is our K-State wheat pathologist, is going to kick us off and get us rolling to understand the biology of these diseases. And then Anthony Zukov, who is one of our entomologists in Garden City, is really going to talk about the biology of the wheat curl mite. So we understand better how they move, how they reproduce, why they can cause such a big fuss as they did this last year. And then we're going to switch gears just a little bit, talking about controlling volunteer wheat with herbicides. Dr. Sarah Lancaster is our state weed specialist, and myself, and we're going to kind of go through control measures that we can do to get a handle on the volunteer weed out in the field. Finally, Romulo Lolato, who is our K-State wheat specialist, is going to talk about varieties and how tolerant or resistant they are to these mosaic viruses and other management things that we can go along with helping handle the mosaic virus pressure. So at the end of all of this, we're going to have kind of an open Q&A session. So make notes as we're going through of questions that you may have. You can be ready to put them in or ask the questions of this whole group that we're going to be, all of our speakers that I've mentioned so far. In addition, we'll have Logan Simon, who is our Southwest area agronomist, Tina Sullivan, who is our Northeast area agronomist, and Lucas Haig, who is the agronomist in charge at the Tribune Experiment Station. So that's kind of our schedule for the night. And then if you are ready to ask a question, which you can do at any time, you can put it in the Q&A box. So down at the bottom of your screen, you'll see a little box that says Q&A. Click on that, it will pop it open, and that helps us track the questions as they are coming in. So put any subject matter questions. So if you don't understand something that you're seeing in a chart, or we talked about something, but we need to go more in depth, put that type of stuff in the Q&A box. We also have a chat box on there, and that's if we are having some technical difficulties. So if you can't hear something, can't see something, we have a couple of behind the scenes folks that are helping us out with that. Kathy Gale, who is our person who handles all of our webinars, and she's our agronomy e-update editor. Kathy's in the background. We also have Sandra Wick, who is the extension agent in Post Rock District for the crop production area. So they are both in the background. They're going to be the ones kind of handling our chat box questions or any technical things that are not working. If all else fails and you can't get through to any of us, you can go ahead and just send me an email and with a question that you have, and I'm happy to get that asked as we go through that. Finally, we do have certified crop advisor credits. So CCA credits are available. We've applied for them. And so you can email me right now to get your name on the attendee list. And then at the end of the webinar, email me again. That way we know you've been here for the whole webinar. We know you've been participating. Include in that email your name and your CCA number. That way I can get it on the list so that we can get credits for you. So go ahead if you're attending for CCA credits, go ahead and do that right now. Sandra is going to put that in the chat box, so you don't have to try to write this down real quick and get it, but you can copy and paste out of the chat box. So with that, I think we are ready to kick off and get rolling here. So we have Kelsey Anderson Onofre, who's our K-State wheat pathologist, spent a lot of time this summer looking at wheat that was showing mosaic symptoms and things that were going on out in the field. And so Kelsey's going to kick us off with talking through the biology of these diseases and really kind of get our feet under us so we understand the basics, so then we can understand the management behind it.
Yeah, absolutely. Thanks, Jeannie. And thanks everyone for joining us tonight during your evening, right? For taking some time out to visit with us. If you're here, you probably understand that this was an important issue in Kansas this year. Jeannie, can you see my screen? Can you see the slides there?
Looks good.
Excellent. Okay, well, I'll kick us off. So Anthony and I are going to tag team this first piece of the discussion. We're going to talk a lot about the biology of the virus and set us up with some information about the wheat curl mite itself, right? And then we'll kind of end with some management, and that'll lead into Sarah's talk about herbicides for volunteer wheat control. So wheat streak mosaic, right? That's why we're all here. This was a fairly widespread issue across Kansas this year. It really did vary in terms of severity, but we saw symptoms that looked similar to this in most wheat fields in Kansas, even at low levels. And in many areas, it was very severe, right? There are a lot of things that can cause yellow wheat, right? Nitrogen deficiency, for example, lots of other stresses. Wheat streak mosaic virus is characteristic in that it causes these yellow streaks that form between the veins, but also what we call a mosaic pattern, right? So you can see a kind of a blotchy green and yellow appearance, and that's very classic for a wheat streak mosaic virus, right? Now, a couple of things. We sometimes get confused with wheat streak mosaic virus. So very early stripe rust will sometimes form just very faint streaks, and that can be confused with wheat streak, right? So we had that question a lot this year. You'll see a small streak on the plant. Maybe you think it's early stripe rust, and that might trigger a fungicide decision. And in many cases this year, that was actually early wheat streak mosaic, right? It didn't progress into the spores that we typically see for stripe rust, right? We do see symptoms vary across variety and also based on the composition of viruses that we see in different plants. So for example, sometimes these symptoms appear more white and sometimes they appear more yellow. That varies a lot based on again, the variety and the viruses. So in very severe cases, right, this is a bad case of wheat streak mosaic virus. We also saw worse cases, right, where we see extreme stunting, right? So usually that's an early infection. We see extreme plant stunting. Sometimes in those cases that we don't see the plants heading out. And then, of course, we see the plants more splayed out, and the yield loss can be substantial, right? Sometimes we'll see that symptom appear towards the edge of a field where we had a source nearby, and sometimes that's just gone clear across the field as those mites have worked their way through the landscape. Again, we see a lot of variation in symptoms, so sometimes we do have plants with height. We'll talk about that a little bit more, and that really depends on the timing of the virus infection. So what are some of the pieces that went into the outbreak in 2025, right? We'll touch on those a bit here, but one of the key pieces really was the weather in the fall. So here in this graph, we're looking at deviation from normal from September 1st through November 30th here. So that's really through our fall period. And you can see that red indicates that we deviated from normal. So we had warmer than normal temperatures really straight through that 3-month period. Now, why that's important? This period is really critical for mite development. And when the weather is warm and we don't get a cold spell, those mites don't slow down. So they were able to really work their way across the field for extended periods of time and build to high levels and cause some of those very severely affected fields that we saw this year in Kansas. That's a piece of the puzzle. And Anthony will touch more on, those exact temperature requirements that really drive those mite populations up. When we're thinking about wheat streak mosaic, it's really important that we think of it as a complex, right? So this isn't just one virus, it's actually a group of viruses that infect together. And when they're present together, they can cause more severe damage. We'll talk about that in a moment. Now, why it's important to think of this as a complex is that they cause very similar symptoms. So it's almost impossible to tell triticum mosaic virus apart from wheat streak mosaic virus. And these viruses are all moved around by the same vector, that tiny wheat curl mite. So here in Kansas, we have wheat streak mosaic virus, triticum mosaic virus, and high plains wheat mosaic virus. Again, all three of them are moved around by that tiny wheat curl mite. They all infect wheat, and they can cause some pretty severe symptoms. Now, here's some kind of common questions that we get associated with those curl mites and these viruses. So a key piece here is that the same one mite, or the same population of mites in a given field can actually have all three viruses at once, right? And it's very common for us to receive plants in the diagnostic lab to test plants that have all three viruses. We'll talk about why that's important in a moment. Some other just basic facts about how these viruses work. Unlike some other viruses that are moved around by insects, these mites acquire the viruses, or as they're feeding, they pick up the viruses very quickly. So they only need to feed for a very short period of time to pick up those viruses and then transmit them to the next plant that they feed on, right? And they carry the viruses with them in their bodies through their whole lives. They can't move them to their offspring, so they don't move to eggs or offspring, but they do keep them for their entire lives. And any plant they feed on in that time will become infected. That's one of the things that makes this so difficult to manage. There are several things. So we get a lot of questions on what drove the yield loss in any given field that had wheat streak mosaic virus, right? One of the very important pieces of that puzzle is the composition of viruses in a given field. So if you have, and we'll talk about this in the next slide, more than one of those viruses present, you'll probably have a higher chance of yield loss. The other piece is the variety, right? So the variety choice, although we don't have perfect varieties, and Romulo will talk about this in a moment, the variety choice really can influence the amount of yield loss that we experience. The timing of infection is also critical. Earlier infections result in higher levels of yield loss. And of course, as we've been talking about, that weather and how that influences that mite relationship and the virus movement in the plant is a really important piece of the puzzle, right? And so in a year like this, we get a lot of fields where all of those puzzle pieces came together perfectly and we experienced high yield loss. We talk about synergistic infection, but here's some really good data from colleagues in Nebraska showing how those co-infections can really result in reduced yield. So here, for example, is an unaffected plot or an uninoculated plot, right? We're having yields kind of shy of 80 bushels per acre. And you can see that dramatically drop when we have a single infection of triticum mosaic virus versus a single infection of wheat streak mosaic virus. But what I think is really interesting here, and we've seen play out a lot in this season, is when we have those co-infections of wheat streak and trim V, the yield drops off dramatically, right? They work synergistically together, they help each other out, and they can really drive down the plants, right? And again, we saw that play out this year. I threw this graph in here just to show that we do get a reduced 1,000 kernel weight with wheat streak infections, right? We get some more lightweight kernels in affected plants. So that's one piece, right? The composition of viruses. When we have two or more of those viruses, we do have higher yield losses. The timing of infection is also really important. So often we see these early infections, right, in this maybe three-leaf period in that fall, right after planting, right, or the 5-leaf phase, early spring, or even through jointing. And some good research that looked at that, again, out of Nebraska, showed that those early infections really result in higher yield losses, right? So the later the infection happens, the less severe the yield loss is going to be. And that was repeated over time here with some consistent results showing that earlier infection is really important. Now that brings up a pretty important question, because often we get fall infections that can be pretty severe, but we don't see symptoms until the spring warms up after green up, right? So the mites feed, but the weather's too cool and the plants are too small, and that virus needs to work its way into the roots of the plant. So that virus almost remains dormant in the plant until the weather warms up in the spring and we hit those 70, 80-degree temperatures. And that's when we start to actually see the symptoms, even though the infection took place back in the fall or maybe the early spring, right? And I think this year we were surprised or caught off guard by a lot of those symptoms showing up when the weather warmed in Kansas in April, but we did have those more severe yield losses, which indicated an earlier fall infection. So again, there we have two pieces of that yield puzzle, right? One piece is the composition of viruses, and that second is that timing of infection. So, I'm pulled in the diagnostic lab data just to show that we had all three viruses pretty active across the state this year. And we did have a lot of dual or even triple infections, right? So again, this is data of samples that came into the diagnostic lab, right? So this isn't a survey. This doesn't include fields that we visited or virtual diagnostics or anything like that. These were just samples that came in through the diagnostic lab, and that number is the number of positives for wheat streak mosaic in each county. So you can see pretty much across the state, these were the positive samples. Now, I know we had fields in the white counties that don't have represented samples here, but they just didn't come through the diagnostic lab. But what's interesting here is this is that same map for triticum mosaic virus. So the last one was wheat streak mosaic. This one is triticum mosaic. What I want to point out here is we had 144 positives come through the lab of triticum mosaic. A lot of these were in co-infections with wheat streak, but not all of them, right? So we did have some single infections of wheat streak, of triticum mosaic come into the lab. But I think an important piece here is to point out that we have triticum mosaic pretty much throughout the state, or the major wheat growing parts of the state. Maybe 10 years ago, we had triticum mosaic that was more limited to fields in western Kansas, but this year it's pretty clear that we have very common infections of triticum mosaic statewide, and this is something we need to be paying attention to and taking into account when we're choosing varieties. High Plains wheat mosaic virus was also unusually active this year. We had more diagnostics positives, I think, than ever in this year, and pretty much across the state, right? And again, we did have samples with all three viruses present. So we're going to switch gears here, and we've been talking about the viruses. We're going to switch gears here, and I'm going to bounce things over to Anthony to talk a little bit about the biology of the mites.
Yeah, thank you, Kelsey. So yeah, good evening, everybody. Yeah, we'll jump over to the actual vector itself. Just kind of hit on some basic biology and some dynamics about how it disperses and things like that. But one of the most challenging things I think when it comes to this particular pest is its reproductive capacity. So this mite reproduces extremely quickly, as you can see from this slide. We'll go through some of that. So we can get a new generation of wheat curl mites in as little as a week, 8, 10 days. So that means from egg to reproductive adult, we can have, every week almost a new generation of mites that are going to be laying eggs, reproducing. And they do really well between 70, 80 degrees. So this is like a landscape level pest. I always like to say that. And no matter what we do to manage this pest, we could do the best cultural things. We could be prepared for it, but we cannot control the weather. And so when we have really great favorable weather for this pest, it reproduces fast by itself, but when the weather is really great for it, it's going to reproduce even faster. So there was mention about, we have increased kind of warm falls going on lately. That's going to allow the mites to be more active and continually reproducing through that extended fall. And so we have multiple seasons like that, and we're going to have mite populations build up in over summer and over winter. So they can tolerate really cold temperatures. So even in warm winter days, wheat curl mite can be active in fields. So they're not going to be killed off by super cold temperatures for the most part. So they're very resilient, even though they're kind of delicate at the same time. But as you can see there, you know, especially in the red, I mean, we could have billions of mites in a field at any one time. So when we have really good weather that's favoring mite reproduction, uh, we have years like this where we had wheat streak mosaic blowing up in last year. So um, and they can survive, multiple days outside of a host. They're delicate, but they can survive a few days, and they do move very easily. So I think there's a slide coming up where we have our maybe some movement. So, oh, well, okay. We'll just go with the appearance. So you'll be hard pressed to see a wheat curl mite with a hand lens. So we have on the upper left, we've got a um, microscope photo of a wheat curl mite. On the upper right, you see that's kind of like a dissecting scope image. So they're very small, cigar-shaped, tiny little mites. The bottom left, you'll have their piercing, sucking mouthparts. So these mites are going to be destroying individual cells on that wheat leaf. And when they're feeding, they're going to be causing the plant to respond in a way that causes curling. So when you're dealing with wheat curl mite, they get their name because they're going to cause that leaf edge to curl inward. So the very edge of the wheat leaf is going to be curling in. So if you're comparing that to um, Russian wheat aphid, for example, Russian wheat aphid causes leaf curling, but it's going to cause the entire leaf to curl. Whereas wheat curl mite, it's just going to be the very edges of that leaf to curl in, and you don't even notice that. You really won't notice wheat curl mite problems until you actually start to see, um, you know, the mosaic virus complex. So that's what they look like. They're very small. And the actual physical damage they do is minimal. It's just the virus that's going to be the issue. But the dispersal is important to discuss. So these mites, they get around really, really, really well, but they don't fly. So they're not flying around. They're moved by the wind. Um, they could go multiple miles from a dispersal point, carried by the wind. Like I said earlier, they are kind of, delicate organisms, but they can survive for a few days off of a host plant, especially when they're carried up into that wind column and up in upper atmosphere where it's cooler. They can survive a little better. It's not so hot, but they can get dispersed very, very far by the wind, and that's how we end up having wide scale problems. Like I said, this is a landscape level pest because they're moved very easily by the wind. So it's important to understand the biology and dispersal of this pest because they're going to be moving from that volunteer wheat, right? So if we have that volunteer wheat between harvest and planting, we're going to have potential for that mite rain, right? So as you see here on this slide, we can have large scale dispersal of these mites as harvest kicks in. They're going to be moving to the tops of the plants and dispersing and getting into that green bridge zone where we have maybe volunteer wheat or early planted wheat, and they're going to be reproducing that whole time from harvest through new planting time. And so this is a great slide just to show you, like I was saying, the mites build up at the end of the season. They'll migrate towards the top of those plants, and they will be moved very easily by the wind to whatever volunteer might be there or other alternate hosts, which I think someone will talk about alternate hosts later on. But that, yeah, they disperse quite widely, simply leaving those fields that are dying down before harvest. And it's important to understand that, so when you have a source of mites in maybe some volunteer wheat or a field that's drying down, their radius of impact is going to be a lot bigger than where they are originating from. So you see that red circle where you have, a source of potential mites that are going to be leaving that field. They're going to impact a larger area than what they came from, because remember, their reproductive capacity is really high to begin with. And if we have a fall that is, conducive to making the mites happy, making volunteer wheat happy, alternate hosts happy, the population is just going to amplify. And it could get really large really quickly going into planting season for wheat. And typically when you have sources impacting nearby fields, it'll start at the field edges. You'll see impacts from wheat chromite and wheat streak mosaic at the field edges as the wind blows them into those areas. Um, and you could have areas in the middle of the field that also kind of erupt and start showing symptoms. And that's just a kind of a factor of how the wind is moving that mite population from the edge of that field. So the edges is where you'll start to see the biggest impact, but you can have islands, so to speak, that erupt in the middle of a field. Um, so you can kind of expect in bad years like we have been having, you will see some center of the field type impacts, not just the edges. Right. And then so again, so volunteer wheat really is something that we're talking about that right now, right? So that is one of the biggest concerns going into planting season. Volunteer wheat is the um, biggest driver of supporting wheat chromite populations in our landscapes. There are other hosts, but they don't do as well on those other hosts. But again, if we have these large areas of volunteer wheat, that volunteer wheat area, the impact from that is going to be a lot bigger than uh, it's going to be a lot bigger than what we have for you might think a small amount of volunteer wheat is no big deal, but you're going to have a bigger impact, as you can see. So that slide here just shows you a small amount of volunteer wheat can really make a big negative impact on the landscape. And yeah, fine. Yeah, just so we get this question a lot. So what can we do to control the mite, right? Fungicides, for one, will not have any effect on the viruses because they're viruses. And insecticides and miticides are not recommended to control wheat chromite. For one, they might reduce the population, but the insecticides and miticides are not shown in any studies to reduce the incidence of viral load in your fields. So do not choose to use an insecticide, a fungicide, a miticide, a seed treatment to control wheat streak mosaic or related viruses.
Awesome, Anthony. And yeah, that's a great point. All the data is pretty clear. You might kill some mites, but the time that they need to feed is so short that they've already fed and they've already transmitted the virus, and then you'll have new flushes in. So really not practical to use insecticides. So what can we do, right? So really everything we can do happens between now and fall planting, right? So the bottom line is, volunteer wheat management, and we'll talk about some updated recommendations for volunteer wheat management. I'll touch on that first, and then Sarah and Jeannie will go into more detail about that point. choosing a right variety, Romola will talk about that, and situational awareness. So knowing where we are in relation to surrounding cropping systems is a huge piece of this puzzle as we gear up for fall planting. So we're introducing the concept of wheat-free windows, right? So some updated guidance for volunteer management. So I'll introduce these quickly. Again, Sarah will touch on them in more detail, but these are wheat-free windows based on our recommended planting date zones, our recommendations for planting dates. So here, for example, in Northwest Kansas, we start our recommended winter wheat planting window on September 10th. So we're recommending keeping that area wheat-free or volunteer wheat-free in the 30 days prior to planting, starting on October 10th. Those wheat-free windows adjust a bit based on recommended planting date through to here in Southeast Kansas, where we're looking at September 5th to October 5th, right? And this period is really a time where we can make sure regionally that our mite levels are reduced, right? So here, this is a little bit silly, but we really want to knock down those mites, right? Bring down those mite populations, lower the mite load moving into that period. And we also want to have some synchronization of that period of winter wheat control, right? We don't volunteer wheat control. We don't want to go in and control our volunteer wheat at some point in August, kind of bump our mites over to our neighbor who's controlling their volunteer wheat a couple weeks later, right? And hopscotch mites in the landscape, right? So a key here is being a little more focused on this period, right? It'd be ideal if we can control volunteer wheat all summer. That's not practical, right? So we are recommending that volunteer wheat be completely dead prior to the start of the regional wheat-free window. That's a mouthful to do twice today. Where possible, we want to avoid planting that fall wheat crop during that wheat-free period. We also need to be mindful of other winter cereals. I'll talk more about these in a moment. But these winter cereals like rye and triticale planted early can be that bridge for those curl mites to make their way to the fall established wheat crop. This regional break in volunteer can give us a nice clean break for the winter wheat crop and time for those curl mites to die off. Now, a common question we get is about emergence after that window, right? So what if volunteer wheat emerges right at planting or right before planting, right? That would be less concerning because that volunteer wheat just simply had less time for those mite populations to build up, right? So again, this is a kind of a coordinated approach and a more focused approach on a window to have volunteer wheat completely killed off in key regions in Kansas. So I'll just touch quickly on some of the role of other grass hosts in the landscape. We get this question a lot too. And so there's a few different factors that play into how important some of these other hosts are outside of wheat and volunteer wheat, right? And when we think about the risk of these hosts, we have to think about all of these factors, right? So one is how well the curl mite reproduces on any given host. It does its best work. It is the wheat curl mite. It does its best work on wheat and probably barley. It reproduces its fastest. That's the host that it really enjoys. And then it kind of goes down from there, right? So other hosts, maybe it reproduces a little bit slower, right? Those numbers that Anthony talked about, they go down on other hosts, right? They just won't reproduce as well. But it also matters how susceptible that other host in the landscape is to the virus. So if it's not susceptible to the virus, that plant can't transmit the virus to the curl mites. And then the timing of when that host is present or when that species is present is really important. So it does matter, say, if the crop is there or the other species is there right at wheat harvest when we have that mite rain happen, and if it stays through the summer so those curl mites can build and it's still there and still green right at wheat planting, that would be a higher risk crop than, say, a crop like jointed goat grass, which follows the wheat kind of timing and typically dies off around wheat harvest, right? So even though it's susceptible, it wouldn't be as high of a risk. And we want to know again how those periods overlap with the wheat crop. So is it there at those key times when the mites are leaving the wheat crop? And how abundant is it? How much of it is in the landscape, right? If there's a few, say, pockets of a weedy grass out all summer, that's probably less important than a really big area of volunteer wheat, right? We talked about those graphs that showed kind of how big the influence of any given size of a source is on the landscape. So the density is really important there. Again, volunteer wheat, it can meet all of these pieces and can certainly be very high risk. But there are other crops, right? We talked about wheat. Barley can also be a host for the curl mite and the virus. Rye, corn, sorghum. We'll talk about corn and sorghum a little bit more here. Triticale. There are a lot of grassy weeds that can also host the mite and the virus that have been found to be positive for the virus, and we can find curl mites on them. And then there's a list that's a bit longer than this of some of our pasture grasses that also can host this mite and virus. So let me touch on some of our crops. So rye, triticale, barley, and oats all have been well documented to be susceptible to the viruses and also host the curl mites, right? So if they're there in the summer, they can be a high risk for those mites and of course, for the viruses, right? Again, they're at the highest risk if they're planted early or over the summer and they have time out in the landscape to build those mite populations. And if we have winter wheat planted adjacent to them, that can be a problem, right? Now, one important piece is that crops like triticale, they might not show symptoms. So they're asymptomatic. They might look beautiful and green, but they actually have those curl mites and can have that virus as well. We get the question about corn and sorghum a lot, so I wanted to touch on those. Some of the data shows for both of those crops, corn and sorghum, the curl mites, they can reproduce. It's not their favorite crop. They can reproduce okay, but they do much better when the plants are small, right? They don't do as well when the corn or sorghum have, big, fully developed leaves. They do much better on the smaller corn and sorghum plants. And then for corn, there's been some good data out of Nebraska that's shown that they're really limited to the husks at the end of the season. So you can find the curl mites on the husks, but it's harder to find them on the leaves. And when they feed on the kernels, they actually have a toxin that they emit and they cause kernel red streak, right? So sometimes we see that it's a cosmetic problem, but it can be an indication that curl mites were active in that cornfield. Now, some other data from Nebraska has shown that the curl mite populations, they really decline quickly after corn reaches black layer, right? So most of our corn that's reaching black layer before winter wheat is planted wouldn't be of concern. And also those curl mites, they don't move out of a corn canopy as easily as they move out of a wheat stand, right? Or a stand of volunteer wheat. So again, we get those questions a lot, and we can discuss that more. If you have additional questions, feel free to put that in the Q&A. If you want, we offer diagnostic services for these viruses and several other wheat viruses, and of course, all sorts of plant diseases in the plant disease diagnostic lab here in the Department of Plant Pathology. So again, we're always happy to accept samples, and you can submit them through your local extension office there. And if you submit them through the extension office, you do get a discount. So that's always something I like to remind folks. If you have any questions, you can feel free to reach out to myself or Anthony, and we'll take additional questions during the Q&A. But right now, I think I'll pass things over to Sarah if she's ready.
Okay, but while you're switching it over, there were two quick questions that you might be able to answer real quick. The first one was for Anthony. Are there any other major vectors than the mite? Is Anthony still on?
Yes, I'm here. Can you? No, I mean for wheat streak mosaic and that complex of viruses,
Yeah.
no. The wheat curl mite, to my knowledge, is the vector. And
Okay.
Kelsey, you can chime in as well.
Okay.
No other known vectors for those viruses. So aphids cannot move these viruses around. It's a very specific host vector relationship there.
Okay, one other quick question. Are there any natural predators or diseases such as, either bacterial, viral, or fungi that can attack the coral mite?
So the nature of their feeding, how I mentioned earlier, how they'll feed on the plant and the plant responds, kind of curls up and creates their little mite microhabitat. The wheat curl mites are very protected. And so predation and parasitism are going to be extremely low for wheat curl mite. And that's another reason why applying insecticides or miticides are not very effective because they're protected. They create their own little microhabitat. So no, we can't, unfortunately, we cannot rely on biocontrol for this particular pest.
I've only ever seen one study that suggests that thrips can feed on, I don't know, Anthony, we've never talked about this, but thrips might feed on wheat curl mites, but the flip side of that is you don't want thrips in your field, right? So sorry, Anthony.
No, that's a good point. Thrips are very wily and they can get anywhere they want to go. So I could see mites being maybe preyed upon by thrips, but again, not to a degree that we can rely on thrips to help us. And of course, thrips are going to be a plant pest as well. So they're opportunistic. They will eat other insects. Thrips will eat spider mites, spider mite eggs, but they also feed on plants. So yeah, we don't want thrips in the field. So we can't rely on them either.
Great. I'm glad we answered some of those quick questions here. Thank you, Anthony and Kelsey, for jumping in on those. Okay, Sarah, we can see your screen. It's in presentation mode. And so I think we're ready to switch gears just a bit to when we're talking about the management of volunteer wheat to really help kind of suppress the pressure from wheat streak mosaic viruses.
Awesome. So Jeannie, you just chime in as we go through this, and I think we'll spend the next 15 or 20 minutes and it'll be good. So is my audio okay?
Yes.
Okay, good. Okay, so one of the things we talk a lot about in weed management is this idea of integrated pest management. And a key piece of that is just knowing kind of where your weed problems are coming from and knowing how maybe you can prevent them. So just wanted to touch on, Kelsey talked a lot about the temperatures and maybe why wheat streak was more problematic this year than it normally is. But, just wanted to take a moment and think about what causes volunteer wheat, right? And you guys know, head shattering, wind, hail, delayed harvest. We can't do much about that, but we need to know to be prepared, right? Lodging, so we can maybe think about variety selection to avoid lodging. And then lastly is combine setting. We spend a surprising amount of time talking about how to control volunteer crops in the next phase of the crop rotation. And so just, fine tuning some of those combine settings to minimize losses at the header as well as coming out the back of your combine can just be a way to maybe reduce the need to mitigate this problem later. So a lot of the big, emphasis of this program this evening is on changing how we think about managing volunteer wheat. So moving from I'm going to control my volunteer wheat 2 weeks prior to planting, and you're going to control your volunteer wheat 2 weeks prior to planting, to we want everyone to aim to control their volunteer wheat for this 30-day wheat free window in the region, just based on what is the optimal planting date in that region. So Jeannie, I thought this would be a good place for you to kind of drive for a little bit.
Right. Yeah. So we're looking at, if we're in Northwest Kansas, we're looking at doing some spraying at this point or in the next week or so to really get our volunteer dead and dry before this wheat free window. And so we have for Northwest Kansas, the wheat free window from August 10th August 10th to September 10th. You know, our optimal planting time for the northwest corner of the state is September 10th through September 30th. And so when we are seeing those type of windows for planting, then we back up a month ahead of that. So really extending that period out. And what Kelsey mentioned is that we're trying to get away from the fact that we're hopscotching back and forth across, basically moving the curl mites around to wherever there is a source of volunteer for them to be able to get established and continue their reproduction. So you'll notice as we move southeast across the state, those dates are shifting as we get down there. If you look at the optimal planting date window map that we have for K-State for across the state of Kansas, we have that same stair step as you go down across the area. Clear down to southeast Kansas, we're looking for that wheat-free period from September 5th to October 5th. The optimal planting window there is October 5th to October 25th. And so really we're kind of making that arch across the state. So you know, what we're really trying to get away from is that we are not going to have volunteer. So those curl mice are just moving around to the newest, best source of volunteer or other hosts for them to be living on. And we, Anthony mentioned, you know, we talk about 4 days of them when they die after living off the plant. We usually talk to 7 or 8 days thinking about that. And now we're extending that period that much more. What this also means is the struggle, because I know when we get moisture in western Kansas and we are close to September, in September, we want to plant wheat because we've got moisture to get a stand. I totally understand that. And so what we're asking folks to do is to just back that planting window back up. We're not going to plant wheat on Labor Day because we want to stay out of that period of when the curl mites would be, you know, a good place for the curl mites to show up on that. So we're moving back into those optimal planting windows and having that longer period of time where we have no volunteer out in the field and growing. The other thing I think about when we're talking through this is actually knowing where we have volunteer wheat out in the field. And so I also think that that means we need to actually get out of the pickups, walk out and look a little bit closer, move the wheat residue around and look to see what is actually under that area. And because I know right now we're driving by, and while we may see Palmer amaranth out there, there's not that many. We're like, okay, our wheat stubble is in pretty decent shape. What I'm asking you to do is go out and look closer. So in many cases, we're not cutting wheat stubble at the 3-inch or 6-inch height. We are cutting wheat stubble either taller or we have stripper stubble. So we actually need to get out and look down in that stripper stubble or high cut stubble to see what is coming up for volunteer out there. And I will tell you, I cut wheat for 3.5 weeks this year because we kept having moisture, which is excellent. We're so happy to have rain. We'd also like to harvest wheat, but when we were finishing wheat harvest, I was driving past fields where there was volunteer wheat coming up in it. And so I want everybody to be aware. I've seen volunteer out. We've been catching some of these showers. It's actually just showering in Colby right now because I'm sitting at the office for this webinar. And so great conditions for getting our volunteer wheat started, which also means we need to be on our game for getting volunteer wheat controlled. And so go out, look close in the canopy to see what we have for volunteer wheat that's kind of down in that residue, to know what is carrying on. Check the edges of the fields, check areas where you think you might have had some wheat laying down. You know, I always check on the edges because I figure as we're turning at the edge of the field, we're having anything that might be around coming out. So check in those pockets to see what you're finding for volunteer wheat, but get out and actually move residue to see what's down there.
So if you know you have volunteer wheat, we know we're going to need to spray. So I organized this list of herbicides based on some things that are in the chemical weed control guide in the fallow section, but I organized them by herbicide group. We're actually not going to talk about them in that order. We're going to hopscotch around just a little bit. So I think we should start by talking about glyphosate. So that'd be the third line on our table here. As we think about controlling grasses, including volunteer wheat, glyphosate is definitely sort of the easy button there in terms of the answer for a recommendation for control. Glyphosate provides excellent control of volunteer wheat. Two downsides to relying only on glyphosate. One, glyphosate has no residual activity. And secondly, glyphosate takes a long time to work, to effectively kill the plant. And so we're going to look here in just a minute about why that can maybe be problematic. So the next group we'll talk about then would be some of the group 14 products like Reviton and Sharpen. So those are contact herbicides that require good spray coverage, require the addition of both MSO and a nitrogen source. They by themselves do not have good grass activity or great grass activity. However, if we add a product like that to a glyphosate application, we can speed up the kill of that glyphosate. So that could be something to consider if you want to try to kill your glyphosate a little bit faster. Also realize that Sharpen has a little bit of residual activity there. So speaking of residual activity, the next things I think we should look at here would be atrazine and metribuzin. Those are probably the two most commonly kind of thought about residual products here of this list. They're both group 5 herbicides, which means they synergize paraquat applications. And we'll talk about paraquat a little bit more in just a minute. But those could be options to provide some residual activity. Metribuzin will be more suppression than true control, but it still can be helpful. And then the last two items on the list there, we have products that contain flumioxazin. So that's the active ingredient in Valor. And then products that contain sulfentrazone. So that would be the active ingredient in Spartan. So lots of options for brand names for those two products. They're two group 14 herbicides. So they're PPO inhibitors, but they're going to provide some contact activity as well as some good residual activity for these systems. In addition, they're going to be good for kind of, Jeannie mentioned the pigweeds and the kochia, things of that nature. So those are some options for residual control. And then, last but not least, in a discussion about herbicide recommendations for wheat stubble is paraquat. Paraquat is a contact herbicide. It does require, excellent spray coverage in order for good activity. I did a presentation earlier this afternoon and described paraquat as having, good, not great, better than fair, but not great. So good grass activity if you can get the spray coverage. Things we need to think about for spray coverage there would be spray volume. So thinking 15 to 20 gallons per acre in all likelihood, but also thinking about droplet size. So, we want to hit that sweet spot where we're getting good spray coverage, but not having a lot of drift. And so typically that's going to be somewhere around that coarse droplet size. Okay. So, I'm talking again this afternoon about Liberty applications. That's our target there as well, that coarse droplet size. So paying attention to nozzle selection and operating pressure to make sure we're not drifting too much, but also getting good coverage. The other thing about that droplet size piece, Jeannie talked about tall wheat stubble, stripper stubble scenarios. We need larger droplets in those scenarios in order to get good penetration of that canopy. It can be tempting to say, I want to force my application down to the ground through that canopy, so I'm going to increase my pressure on my spray application. That's actually just going to create more fines, more driftable fines, and smaller droplets. So the thing to do is to keep that pressure low, use larger orifices, and get that kind of coarse droplet size to get good coverage and good canopy penetration as well. Jeannie, what did I miss on that slide?
Think we've covered a lot of stuff. I just mentioned that as I'm talking to guys controlling just in their wheat stubble in general, flumioxazin, sulfentrazone comes up in discussion. Both of these are products that we are using more often than we have in the past in many cases when we're thinking about using them to go out early to get a handle on some kochia. And so we're weaving these two products, specifically the Valor, the flumioxazin, into more portions of our rotation. And I always think it's important that we think about making sure that it's effective use of these herbicides so that we are not selecting for resistance type of situation. So as we're using Flumi, I think it is an excellent product. I also encourage guys to sit down and actually look through their whole crop rotation and look how many times we're putting the flumioxazin in there to see if maybe we should be picking the most effective times or the times we need it the most, because we do have some other options in this portion of it. But it's excellent. It does a good job holding things back. It has some activity on grass. It is a contact. And so yes, I just think we need to understand where we're using these products and where they fit the best.
Very much appreciate that comment. Good catch, Jeannie. Okay, so I mentioned when I was talking about the glyphosate applications that, you guys know for the most part, probably glyphosate is going to act very slowly. It could take a week, 10, 14 days to completely kill the weeds after the application. What that means is that volunteer wheat plant that we sprayed with glyphosate is going to act as a host for the mites longer than a volunteer wheat plant that we spray with a contact herbicide, in this case, paraquat. So these are some data that were generated at K-State a number of years ago, but they demonstrate that the longer it takes to kill that volunteer wheat plant, the longer it's going to serve as a host for those wheat curl mites. One of the other data points that they looked at in this paper was the fact that in addition to just more mites in general, when we kill the wheat plant faster, we also reduce the number of mites that are ready to migrate to other plants. So we slow down that migration process, or we can kind of cut off that migration process faster. So, doing things to get a faster kill will give us a longer weed-free window. It's hard not to slip up and say weed-free window after years of training as a weed scientist. Another thought that comes up in this conversation is that tillage can reduce mites quickly. Now, tillage does not fit into everyone's operation, but it does fit into some operations. So these are some data that were generated by folks at the University of Nebraska. And really what they're showing here is that by 7 days after application in tillage, the wheat curl mites were essentially non-existent, okay, in those plots. But that's implying that you have a tillage pass that is effectively killing the wheat. So you may or may not recall the photo that was on that first slide that I showed this evening. I actually snapped that picture this morning while I was driving. Well, I wasn't driving when I took the picture. I pulled over and then I took the picture, but I snapped that picture this morning. And I think you could see in that picture that that field had been worked. I'm going to guess probably with an undercutter. But the volunteer wheat, it was a carpet of volunteer wheat. Okay, so the volunteer wheat had regrown. They'd had lots of rain in that area. So tillage might in some situations be a short-term fix, but if you get the moisture, it's not going to hold on control. Jeannie, what other thoughts did I miss on that one? I feel like you have some good thoughts on tillage or lack thereof in crop rotation.
Yeah, and I think through tillage as a method of this, I totally get it. I also think about the western part of the state and how important wheat residue is in the whole cropping system. And so, I have a hard time with tillage that I think is very effective in our wheat stubble, because we want our wheat stubble standing to help catch snow this winter so that we can use it to put our summer crop back into. So, that is one of the concerns I have with doing the tillage as part of this. I stop and think, like, today it was 100 degrees, had some breeze to it, would have been a great day if we are controlling volunteer wheat with tillage. The next couple of days are going to be a little cooler. We've had some rain, probably not as effective on getting control with that. And so I have tillage can be very effective. Make sure it, in the whole scheme of things, of your cropping system, it is the right fit for you.
One last thing to think about here in terms of weed control, and that would be volunteer weed control in double cropped fields. So in some ways, these are going to be an advantage to us, right? Because we have that crop out there, theoretically, hopefully shading out weeds, including volunteer wheat, but we can have volunteer wheat sneaking in to our double cropped fields. So, you know, again, one of the easy button answers there is if you're in double crop soybeans, which would be the most common double crop around, glyphosate and glyphosate tolerant soybeans would be the easy button there. But again, no residual control. So you control this flush, you get that rain before the beans fully canopy, and there you are, you're going to have to make another application at some point. Another thing to think about here, this is a good fit for metribuzin. At the time of soybean planting, and we can push our metribuzin rates if your soil conditions allow, or soil characteristics allow, pushing that metribuzin rate up to, you know, 10 ounces or so to get some greater suppression of the volunteer weed, work on some of those pigweeds and other things that we struggle with, and still have good crop safety in our soybeans. The next item on the list would be our ACCase inhibiting herbicides. These are our group 1 grass killing herbicides. So if you're in a broadleaf crop, say maybe I passed a field of double crop sunflowers today. So maybe if that's your field, you know, something like a clethodim type of product, like a Select Max or Section 3 could be an option, or a product like Assure or another quizalofop containing product. Another double crop that we see quite a bit of would be types of sorghum. So we do have double team grain sorghum that is tolerant to quizalofop. So that could be an option in a double team grain sorghum setting. The iGrow system, where we can use Emiflex. We have both grain and forage sorghum hybrids available in that system. But for other sorghums that are not herbicide resistant traded, atrazine is going to be our go-to there for some residual activity to keep that volunteer wheat down. Jeannie, other thoughts on volunteer control and double crops that are kind of unique to your part of the world?
Nope. I think the most important thing is to get out and look under the canopy to see what may be coming up out there. I think many times we are not paying close attention to some of that. And then in some cases we can have a pretty good carpet out there and not even realize under the canopy on some of this.
Very good point. Okay, you want to take the conclusions, Jeannie?
Sure can. Okay, so our volunteer wheat should be controlled for 4 weeks. So we're talking a month prior to planting wheat in our region. So a change from our 2-week window to a month to give everybody a chance to get things under control and hold it for a little while so that we are not just moving mites around the area from one field with volunteer to the next. Consider how long it takes for that volunteer wheat to die. And so we want dead, dry volunteer wheat when we're thinking about our herbicide applications. Glyphosate, we know, takes a little bit longer, several days, depending on environmental conditions. I always get the question of how long does it actually take to die? And I always kind of joke and I'm like, well, it depends on what Mother Nature is doing with this. And so I always say, well, we're going to plan on 7 to 10 days, and it may be longer, maybe a little bit faster. But we think about paraquat, that contact control on it. If we can get good coverage on that, we can have pretty effective control pretty quick on that. And we saw the chart where we're dropping that mite population as a result of that. Including a herbicide with residual activity. I always get questions about what if I have volunteer wheat that is coming up as my wheat is coming up. You know, I am not as concerned about that volunteer wheat that is coming up at that point because it's basically the same as our winter wheat when it's all coming up together. I'm mostly concerned about volunteer that's been out in the field for a long time or continually has flushes coming up, a few plants, a few more, and so something with some with residual activity to really help hold that pressure of the volunteer back. And then look beyond our stubble fields to find our winter wheat, our winter cereals that should be controlled. And so look beyond just our wheat stubble, also look down in the wheat stubble, but look beyond it to our double crop, something else that may be out there. Look in road ditches to see what kind of volunteer wheat may be coming up in road ditches. And so just kind of keep an eye out all the way around for where we have winter cereals that can be a host for the curl mites and the wheat Mosaic virus complex. Okay, so Sarah and I are available in a lot of different formats. And so you can see how to get a hold of us via email. We're both active on Twitter and on Facebook. Sarah has an Instagram account. And then if you haven't checked out the War Against Weeds podcast, guys, it's a really interesting podcast to listen to. So a lot of discussion on all kinds of topics related to weed control, not just what we're talking about tonight, but all kinds of different topics, from the basics on chemistry of herbicides clear up to just what do we do after wheat harvest and we have plants that are stressed out there? How do we do a good job getting control on them? So a big topic area on that. Also, the chemical weed control guide, guys, I have one like this all the time with me. So if you can stop by, pick up one at your extension office. If you see me on the road, flag me over. I probably have one in my pickup for you. And so it's a resource that I use all the time. There's one section actually in there that we've kind of emphasized a little bit more this last year, and that is for fields that are going back to wheat this year. There are fallow ahead of that. We have a section labeled fallow, and it's fallow fields that are going back to wheat. So we kind of emphasize that a little bit more than what we have in the past, although it's been in there for a while. And then finally, the K-State Agronomy e-update. We'd love for you guys to sign up to get the weekly update. We just try to send out stuff that we know is important and timely for management on our crops, or things that we're seeing in the field, or things that we're getting questions on. So sign up to get it. I think Kathy just put it in our chat box for folks to sign up to get the agronomy e-update.
Okay, I
All
have
right.
one quick question that came in. If you guys want to answer this, is something maybe you don't really think about that much here in Kansas, but the question was, how would you culturally manage differ for perennial wheat varieties compared to our wheat varieties? And that may be something you want to think about in the Q&A or answer it later.
Yeah, that's a really good question. So we'll put our thinking caps on and come back to that later. So thanks, Sandra.
Okay, and we are now ready to switch gears. So Sarah and I will talk in the background and figure out our answer on perennial wheat. You know, we talk about Kernza as a perennial wheat that we have discussed here in Kansas. So as we're switching gears, I think Romulo will be our next up as our speaker, and Romulo is actually going to discuss how our varieties respond to Wheat Streak Mosaic, to Triticum Mosaic, and actually, you know, how do we pick varieties to help us manage, especially when we're in a situation where there may be a volunteer wheat or other sources around. What can we do on variety resistance or other type of cultural things that we can maybe help minimize our risk?
Very well,
Romulo,
thank you, Jeannie.
I can see your screen and I can hear you.
Awesome, excellent, thank you. Good evening, folks. Great to be here tonight and touch base a little bit with you here on wheat variety selection and perhaps a few other agronomic management practices that we can have to reduce the chances of having another outbreak of wheat streak mosaic virus. But when we discuss wheat variety selection, we know that a few things that go over growers' minds when they are selecting a wheat variety. There are many different things that they will be looking for, right? Different agronomic traits and so on. If we would kind of think about a few of these here, probably the main thing that we're looking for is adaptability, right? Where is that variety adapted to, and what's the yield record that we have for that variety in my region, of course, right? But then a lot of different things trickle down as well that are different agronomic attributes that those varieties will have once you have a list of varieties with good, decent yield record in the region, right? And I list a few of them here in the screen, right? Depending on your previous crop, that might change your variety selection. What's the drought tolerance of those varieties? Are we using them for grazing or dual purpose? And we need those attributes as well. What's their tolerance to acid soils and many other things that you have listed out there in the screen, including perhaps grain quality even, right? Depending if we're part of an IP program or we can capitalize on some protein premiums, as many folks out in North Central Kansas nowadays can. That might be something to consider. But we're focusing tonight here on tolerance to viral diseases, right? And try to understand a little bit more what is out there and what is available to us for us to plant in our farm. But before getting into that, why, right? Why are we focusing on this tonight? And I think that it really is reflected in a couple of different slides that I'll show you here in the next couple of slides. So take a look at this now. What we're looking at here is an yield map that was shared with me in not this year, but in a previous year when we had another wheat streak mosaic complex outbreak. And this was back in 2017, I believe. And this was coming from Horton Seed Services. And what we can see here is you see this purple area in the edge of the field here, and you look at the yield levels on these, less than 5 bushels per acre, right? And as we progress into the field, it increases to between 5 and 16, between 16 and 28, all the way to about 40 bushels per acre or so, as we're a little bit further away from perhaps the source of that wheat streak mosaic virus, right? Perhaps there was a field volunteer that was on this side of the field here, and we can really see that pattern moving up in the field. So this shows us this can be a really, really devastating disease, right? We're talking about almost a complete yield loss in parts of the field out here. This field was in west central Kansas. We go back to same year, but a field in southwest Kansas. This yield map was shared at the time by Edgar Seeds, and we have a very similar pattern here as well, right? Perhaps not as extreme, where we're getting to nearly 0 bushels per acre, but the red part of the yield map here is pointing to less than 17 bushels per acre. And as we progress in the field, that yield level increases all the way to those dark green areas where it's more than 75 bushels per acre. So still, think of how steep the yield loss is here in some of these situations, right? We're going from over 80 bushels per acre in parts of the field to where it was severe, less than 15 bushels per acre, or in the other example, less than 5 bushels per acre, right? So very, very problematic and can result in extremely steep yield losses, right? So variety selection, right, for resistance to wheat streak mosaic virus, which is clearly not depicted in this photo right here, right? This is a photo again from another field with an outbreak of wheat streak mosaic. But the first thing I want to highlight is when we're selecting a variety and we're seeking for that resistance, right, that nothing's bulletproof, right? That's the first thing that we have to keep in mind, because even in varieties that have different levels of genetic resistance that we're going to be talking about here in a second, they are not bulletproof. Now, what is out there and what is available for us here in Kansas in terms of commercial varieties? There are pretty much 2 main sources of resistance to wheat streak mosaic, either resistance to the virus itself or to the wheat curl mite. Right, so let's go into each one of these a little bit more in depth and give a few examples of commercial varieties that are fairly recent and have either one, the other, or both. So thinking about resistance to the virus itself, right? There are 3 primary genes that we know of that can confer resistance to the wheat streak mosaic virus: WSM1, WSM2, and WSM3. WSM1, it does provide resistance to wheat streak mosaic virus, and Kelsey was mentioning before, this is a complex of viruses, right? WSM1 might provide some resistance to the triticale mosaic virus as well, right? Typically, it tends to work better in cooler temperatures, lower than about 69 degrees Fahrenheit or so. But this is derived from a wild relative of wheat. And so when breeders are bringing these into a commercial wheat variety, it can come with some yield drag, right? Whether it is resulting from, lack of cold tolerance to the winters that we have out here, or perhaps maturity is simply not aligned with what we need here in Kansas. So again, it can come with some yield drag coming from some of those other traits that may come together as breeders bring it into a adapted variety to the region. WSM2 would be the most common gene conferring resistance to the wheat streak mosaic virus in our materials here in Kansas, at least. But the thing here is it is conferring resistance only to the wheat streak mosaic virus. So if we have cases, as Kelsey was mentioning, that many of the times this year we were having combined infections of 2 or the 3 viruses at a time, then you will likely not hold, right? We will also see the symptoms out there. Another thing is that it is active under high, cooler temperatures. Some varieties showing a level of about 65 degrees Fahrenheit, below which it works well, above which it tends to be kind of switched off. Other varieties going a little bit further high, about 70 or so. And what this means is that we have a little bit wider protection, right? In the fall, as temperatures start cooling down, or in the spring as it starts warming up. Perhaps the window is slightly larger if we're looking at 70 versus 65 Fahrenheit. And this is derived for wheat, and so from wheat, and so not necessarily coming with potential yield direct here. And finally, the WSM3, right, probably the most recent one out of these 3 genes here, also conferring resistance to both the wheat streak mosaic virus and triticum mosaic virus. The main thing here is that it seems to be effective at higher temperatures, 75 degrees Fahrenheit or more, which even increases our window, our protection window here, both in the fall and in the spring. But again, derived from a wild relative, so bringing these into commercial cultivars, breeders are working on that, but there's many things that need to be figured out before these are released out, again, to avoid bringing some of those characteristics from wild relatives that we might not want in our commercial varieties. So talking about the WSM2 resistance, which again is the most common that we have out there, it can help with, in some cases, even with severe wheat streak mosaic virus infections, but again, it will not protect against those other 2 viruses, TRIM-V or high plains wheat mosaic virus, if they are together in that infection. And if you have mixed infections, as we had many this year, it will also show symptoms. As I mentioned before, right, it will be less effective at higher temperatures. Most of the varieties that we have out here will break down above 65 Fahrenheit. We can cite KS Dallas and KS Hamilton as options that will probably hang in fairly well until about 70 Fahrenheit before they break down. So what this means, that this resistance is less effective at high temperatures, is that if we have some situations where the wheat is planted early for grazing, for example, you will be more exposed to that. Or a very warm fall, as we had this last year. Also, this resistance may not do its job as what we would like it to. Some variety options that we have here in the region, right? Varieties that are well adapted and have good yield record and so on. KS Territory, KS Mako, KS Bill Snyder, KS Dallas, KS Hamilton, Guardian, Oakley CL in terms of hard red winter wheat varieties, KS Big Bowl, and Joe in terms of hard white winter wheat varieties. So these are some of the things that we can talk about here when we're discussing the resistance to the virus itself. Now, as I mentioned, we also have resistance to the wheat curl mite, right? The vector of the virus. This is a gene that we call CMC4, and again provides resistance against that wheat curl mite. And essentially what it does, it will slow down the progression of that disease as the field is getting infected by the disease, right? It's kind of like a less preferred by the mites, so they might try to migrate out of there. And with that, also potentially reducing the risk of very severe disease symptoms in that field. Now, again, if we are in a very high risk situations, right, it's probably still not going to be bulletproof, right? You can see symptoms also in these varieties, even though they will slow down the progression of the disease. If the plant gets infected, the symptoms will be just like on a susceptible variety for the virus itself. So, for example, in terms of just here, right, an example of situation where we'll be very high risk, right? Wheat is planted close proximity to volunteer wheat or to other hosts that are infected with large populations of the mite and virus. Well, a situation like this, might still see quite a bit of symptoms in those varieties. And again, while these varieties will be resistant to the wheat curl mite and slow down the progression of the disease in that field, they will not be resistant to the virus itself, and so the symptoms will still show. This is one example here of a field that we had demonstration plots. So these 2 varieties are in the very same field. You can see a very severe infection of wheat streak mosaic here. On your right-hand side is a SY monument, a check that was being used in this demonstration plot at the time. On the left-hand side, we had bird, which is one of the varieties that has the CMC4. So again, an example here on the same field, it can actually look much better, right, by having that resistance against the wheat curl mite. Even though you see here plants that got infected, they are also showing the symptoms. So again, it will slow down the progression, and it can look like $1,000,000 in some situations, but again, nothing is going to be bulletproof out here. Some of the varieties that are available in the region here, the growers can be looking into options with the CMC4 gene are listed here, right? Many of them coming from the TEM112 family and from Colorado State University as well. Guardian, Canvas, TEM112, Bird, Avery, Langin, Kivari AX, KS Western Star, Whistler, Crescent AX, Incline AX, Fortify SF, TEM115, TEM204, P158 being a few examples here of varieties that have the CMC4 gene and are adapted in the region. And you might have noticed that the Guardian variety appeared in both lists, right, with the WSM2 and the CMC4 gene both. So it will likely provide an improved protection against overall the wheat streak mosaic virus disease. But again, it's not going to be bulletproof because of the weaknesses that we mentioned about the WSM2 gene being only resistant to the wheat streak mosaic virus, not to the other two viruses. So we have been called to fields of Guardian in the past that had fairly severe wheat streak mosaic infection, but looking across the road, you know, it was actually looking like $1,000,000 against a neighboring field that did not have this protection. So it will show symptoms, but it can actually be great compared to a susceptible variety in a high incidence situation. Now, another thing here that we need to be keeping in mind, right? In the very beginning, I mentioned that the main thing that we look at when we're selecting wheat variety is adaptability and yield record in that region. And so one thing to keep in mind is that most of these varieties that we mentioned so far that has the WSM2 gene or the CMC4 gene will be very well adapted to Western Kansas, right? And as Kelsey mentioned before, this disease has been spreading out to central Kansas as well, right? And that kind of creates a gap in the central part of the state there because it hasn't necessarily been the focus of the breeding programs out here. But K-State does have a couple of varieties, KS Mako and KS Buse Snyder, that are fairly new K-State releases that are adapted to the central quarter of the state, actually have excellent yield record, and they carry the WSM2 gene, right? So if you're central Kansas and you're going to be in a high risk situation, you know, probably I'll be looking in one of these two varieties out here. A few other varieties in the central quarter of the state that they don't have any resistance that we know of the mechanism, but every time that we see them in plots, or most of the times that we see them in plots that got hit really hard with wheat streak mosaic virus, they seem to hang in much better. They seem to do a really good job in terms of hanging in compared to their counterparts are listed here too. So it seems like there's some level of background tolerance there by Rockstar, April Prolific, SY Wolverine, LCS Photon AX, LCS Helix AX, and LCS Julep. So these varieties seem to hang in fairly well compared to other varieties when wheat streak mosaic is present. But again, these will appear symptomatic, right? They're not resistant that we know of, and so they will appear symptomatic as well. Another thing here, as we're comparing varieties, right, and this photo here is something that really caught my attention, but Sarah and Jeannie, they were mentioning some of the causes of volunteer wheat early on, right? And here, these two photos were taken from the same field near Hoisington. So this field was split in two, half of it planted with AP Bigfoot, half of it with Rockstar. And as was mentioned earlier here, the harvest was very long this year with a lot of rain, a lot of lodging, and a lot of wind and storms that caused a lot of grain shattering, right? And some varieties, they might hang in better than others, right? In some conditions like these and hold on to the grain better than others. And this is kind of probably what we're seeing here in this photo. You can see a lot of volunteer wheat coming up in the stubble of AP Bigfoot here. Whereas, again, the very same field managed by the same grower, on Rockstar, not necessarily showing nearly as much volunteer wheat out here. So again, differences in variety as well in terms of how much volunteer was going out there as function of their likelihood of going through grain shattering. For more information about the different variety characteristics, right, we have this publication here, MF991. I'm showing here the one for 2024. We are working on the update version for 2025, should come out here sometime next week. And here you have the option of either going more in-depth into some of these varieties and reading their background and so on in these independent panels, or we have a summary table in the back where we have summarizing all of these disease resistance notes that we take during the growing season. And this should help you make a variety selection, not only based on the disease here, in this case, the wheat streak complex, but also some agronomic ratings that we have for these varieties. And finally, just to mention here, but what we're calling situational awareness, right? So just understanding the risk that each one of these fields are going to be exposed to, right? And some of the high risk situations, as my colleagues were actually highlighting here early on, right? Some higher risk scenarios here is, of course, volunteer wheat, right? If we have, as I just showed this photo with hail or shattering, that creates a large amount of volunteer wheat, right? Or perhaps a neighbor who is grazing that volunteer wheat, right? And not controlling. So that for sure being the highest risk scenario, right? But another thing as well that Kelsey brought up is some of these early planted winter cover crops, right? Early planted, and by early I mean sometime in August or even early September, planted triticale or rye, you know, some other cover crop that can actually harbor the wheat streak, the wheat curl mite and wheat streak mosaic, right? So this can again be a source for the wheat streak mosaic virus. I have here the third bullet, summer cover crops right after harvest. So if we're going right after wheat harvest and planting a summer cover crop that has a mixture of grasses and legumes there, perhaps some of those grasses could be problematic and harboring the curl mite or the virus. But not only that, how do we control volunteer wheat in that scenario also becomes a problem, right? Because probably there are no, I'm not sure about herbicide label for cover crops out there. And so again, this could definitely be a concern as well. Situations like what Sarah was mentioning in terms of double crop, milo or soybean or even corn after wheat, right? Where all the volunteer wheat will be there in the canopy and sometimes harder to control. Or fusing your other hosts, right? That might have high mite populations. And on your right-hand side here, there's a publication for K-State that really highlights those different hosts, right? So again, variety selection, if you are in a high risk situation, using these as a tool in the toolbox to reduce the risks of having another outbreak as we had this year becomes very important. And again, because these varieties can look like $1 million compared to their counterparts, and sometimes, you know, delayed sowing as well, right? That can be a management strategy to take if you're seeing that there is a high risk scenario. Perhaps delaying sowing can be an option. Now, how practical that is, as Jeannie was mentioning, in Western Kansas, you have moisture, you're often planting, right? If it's after September. So that from a practical perspective can be challenging, but it is a management that has shown to help improve or reduce, right, wheat streak mosaic infection, again, mostly because we're avoiding those warm fall temperatures when the mites are more active. Very briefly here, we have a number of pre-plant wheat meetings across the state coming up in August. 4 of them are listed out there: on the 11th in Clearwater, on the 20th in Pratt, 21st in Salina, 25th in McPherson, and we're working a few more agents here for more pre-plant meetings. Abilene, Hoisington, and Hayes are the ones that I know of are being worked on. So be on the lookout for those. And on these meetings, we're going to go also in depth in wheat streak mosaic virus, but also more general comments on variety performance, selection, agronomic management, and so on. And here, the wheat RX that is going to go on in Pratt in August 20th is highlighted out there as well. Jeannie mentioned, but agronomy updates, make sure and subscribe to that. We send those, all that information is out every week in your mailbox if you're subscribed to Agronomy Media Updates. Jeannie, with this, I believe I'm reaching the end of my